Thank you so much for joining this presentation today, where we're going to talk about small angle X-ray scattering and how we use this technique to look at protein structure. So small angle X-ray scattering, or SAXS, is powerful because it allows us to examine protein structure in as lifelike or in vitro-like solution conditions. We can also ask questions about the protein structure as a consequence of changing the solution conditions, or looking at it at high concentrations or low concentrations, or we might look at it in the presence of binding partners. So very useful in that sense. Additionally, this technique usually requires only a minimum of sample preparation, and also there are many computational tools to analyze SAXS data, so it can be quite powerful. And in this particular presentation today, the things that we're going to cover are, we're going to look at what information we can get from SAXS. We're also going to look at how it works, and then we're going to look at dilute and high concentration solutions, maybe some ordered things, and we'll give you some examples. Okay, so first, SAXS is a technique that gives you information about the macromolecular size, and it also gives us information about the size dispersity within the solution. And we don't necessarily have to work with solution samples. They can be solid or some other material if you're interested in that kind of thing. And more importantly, they don't have to be light transparent. You just have to have some transmission of X-rays through them. So in addition to size information, SAXS provides us information about the shape of the particle and its morphology. So what does that mean? Well, it means, for example, that we have a very different scattering profile from a virus particle, say, that is loaded with material inside versus one that's empty. That will look completely different, and we can tell that. And we can tell the shape of the virus particle. We can tell the shape of individual proteins in solution. And we can look at whether they're monodispersed. We can also look at if they're monomers or oligomers. So we get a lot of information about shape and morphology. And additionally, SAXS is going to provide information about protein interactions and packing. So are these things ordered packing? Are they disordered? Or for example, are they crystalline? So we can get a lot of information about packing if we want to do so. So what does the experiment look like? Well, of course, we need an X-ray source and a collimation system. And downstream of that, we have a sample holder in which our sample sits. So these are some protein molecules in solution. Downstream of that, we have a detector where we're measuring our scattering data, and generally there's a beam stop at the center of that where the beam actually intersects. Doesn't always have to be the case, but it's there, and usually there's some metal holder, so you might see a shadow from it as well. On the right side, we see an example SAXS image on a 2D detector from a protein solution. And one thing I would just want to point out is that the scattering angle that we talk about in SAXS is called Q. And I mention that because in other X-ray techniques, we often refer to the diffraction angle or scattering angle as 2 theta. Here it's going to be Q. So what do we see? Well, we see the scattering from all the particles that are inside the beam. So that means that if we have monomers or dimers, everything is going to scatter and contribute to the signal that we see. And to process it, it's generally quite easy. So for example, if I drew a line or plotted the intensity from the beam center out to higher scattering angle, then if I did that at a different distance, you'll notice that I would expect roughly to have the same scattering profile. And that's because protein solutions in general that are dispersed are going to exhibit isotropic scattering. If it's not the same at every angle, then we would call this anisotropic scattering. It's probably a material or some other thing, and there are different ways to deal with that. But this is how we deal with protein solutions. Now, what we do is we simply average the scattering profile for the whole image, and we end up with the plot of the intensity versus scattering angle Q, and we generally look at it as a logarithmic of the intensity that accentuate small details in the scattering profile. So you're going to have log I versus Q in any sort of presentations or publications of that nature. So where does the scattering intensity, what determines that? Well, a number of different factors. And we're not going to go over the mathematics in detail. We're going to look at a few of these. So first, the volume of the scattering molecule square contributes to the I with the volume squared. So the bigger a molecule gets, the more it's going to contribute to the scattering intensity. So you can imagine that if you have a disordered aggregate, that's going to contribute greatly to the scattering intensity. And you may not see scattering contributions from very small things. And likewise, if you have mixtures of monomers and dimers and higher order oligomers, then you're going to have the scattering profile of all of these things put together. So that may be helpful in your case, but if you want to look at a single entity, then you have to make sure that it is truly a single monodisperse entity. And you can do that by perhaps doing some dilution experiments, and we'll talk about that in a minute. Another thing that contributes to the scattering intensity is the electron density contrast. And we see the intensities proportional to the square of that electron density difference. And that tells us if there is no difference in the electron density between our protein or molecule of interest and the buffer condition or matrix, then we're not going to see any contribution from it in the intensity. So this tells us also that we could manipulate conditions of the buffer that could improve scattering contrast or cause it to deteriorate. The thing to keep in mind is that the solution conditions definitely play a significant role in the scattered intensity we observe. A couple of other contributors that are very, very important are F of Q and S of Q. And these are the form factor and structure factor respectively. So let's look at exactly where the signal there is coming from. So as we said, the scattering experiment contains information on particle shape and how it interacts with other particles in the solution. Well, the form factor contains the information about the particle shape, and the solution structure factor, S of Q, contains information about the particle distribution or molecular distribution throughout our solution. And if there is no interaction, then the value of this term is 1 for all scattering angles. So we're only going to see information about the shape. So what happens if it's not 1, if there are interactions? We're going to look at a couple of different scenarios, one for attractive forces and one for repulsive. And if we have attractive forces, then we have a form factor for our particles in solution, their shape, and also because of their interaction, the solution structure factor will be greater than 1 at low scattering angle, and it will oscillate about 1 as the scattering angle increases. And the consequence of that is we're going to end up with a product where there's a strong uptick in intensity at low angle. So this is a telltale hallmark of aggregated protein solution, and we'll want to keep an eye out for that. Alternatively, we could have repulsive forces. So let's say that we titrate out all of the ions or salt in a solution, and now the charged amino acids on the surface of the proteins cause repulsion of the protein molecules. In this case, again, the form factor is going to be consistent with the shape of the individual scatterers, but now the solution structure factor will be less than 1 at low scattering angle. It'll approach 1 at some point and then oscillate about 1. And the product of these two will look like something that has a dip at low scattering angle. And again, this is a hallmark of interparticle repulsions. Now, you can imagine in each of these circumstances, the form factor is going to vary based on the entities that are in solution. And if you want to say something about the size and shape and perhaps calculate models, then you need to make sure that your form factor is an example of an ideal and monodispersed protein. So what do I mean by that? So monodispersed means that you have a single population, or all the smiley faces in here, they look alike. They're the same size. They're not interacting at all. This means that we don't have mixtures of monomers and dimers or other oligomers. We don't have repulsion, so there's sufficient ions. We don't have a contaminant of a higher molecular weight or lower molecular weight protein. And additionally, we don't have any aggregation that's occurring. And if we confirm all of these, then we can go on to derive some information, very specific information about the molecule we're interested in. And one way that we achieve this is by doing concentration gradients. So we might collect at different concentrations and ask ourselves, do structural parameters change in effect? So what can we actually get from that? Well, what we can do is we can take the scattering profile and we can look at a number of different factors. And we're going to go through each of these by detail. So first, what we can do is we can plot our scattering data in the form of a Guignay plot. And what that means is that we're going to take the natural log of the intensity versus Q squared. And Guignay in the 30s came up with this law during his graduate work that says the natural log of the intensity is roughly equal to Q squared times RG squared over 3 plus the natural log of the intensity at 0 scattering angle. And of course, this looks like y equals mx plus b. So we gather that we can use the slope of this line to extract information about the size or the radius of gyration in this case. And if we extrapolate to 0 scattering angle, what that's going to give us is roughly the molecular weight. Now, there are some guidelines for the minimum and maximum values we can use. So generally, Q min is, it needs to be less than 1 over your maximum D or your maximum particle dimension. And Q max, it depends on the shape of the molecule. For something that's globular, it needs to be less than 1.3 times the radius of gyration. So if you look at a few examples of this, we see Guignay plots for a bovine serum albumin. And if it's ideal and mono-ideal, then we're going to have a straight line from the Guignay plot. There's repulsion, we're going to see something that looks like a frown. And if there's aggregation, we're going to see that telltale uptick or a smile in our patterns. So that's shown here for 3 different examples. So in the middle, the black for the BSA, that's ideal, whereas we see interparticle repulsion and aggregation with the other 2, blue and red respectively. And this is an example in the literature from 2010. So how would you deal with this? Well, like I said, collect data at several concentrations, calculate the molecular weight and the radius of gyration and ask, is it consistent? Meaning, am I getting to sufficient dilution that I am confident I'm looking at the form factor only? Another thing we can do is look at the shape and by looking at the Raman plot. So this is essentially plotting Q squared I versus the scattering angle Q. And that's going to tell us some information on whether it's folded or unfolded. So a gentleman named Raman did some of this work, and he says that he identified that Q squared I versus Q is increases in intensity and levels out if it's a polymer-like chain, meaning it's unfolded. If in fact that's not the case and the opposite is true where we have folded, then we expect a peak, come back to background, and maybe some additional peaks. Something that's partially unfolded is going to be somewhere in between. And we see another example from the literature for lysozyme. In this case, the black plot shows data and a Raman plot for folded protein, for partially folded protein and its relevant Raman plot. And these 2 are for urea or heated. Both of those are partially folded, and something that's unfolded, meaning that it's in urea and heated, looks like the representative shown on the upper right. So in addition, another thing that we can do is we can also look at the inverse Fourier transform of scattering data. And by this, we're going to look at a pair distribution function. So what's that going to tell us? Well, if we have so here are some representative scattering profiles for different shapes. And if we have a circle, for example, this is the scattering profile we would expect to see for a perfect sphere. And here's some other things. So this is a long rod. It looks like this green line. And if we take the Fourier transform of this, what we end up is some real space measures. So we see the frequency of the occurrence of a given distance versus the distance, meaning that for this green line, there is a high incidence of short distances of about 1, and these fall off as you go down the rod. The sphere looks something Gaussian, but basically the point that I want to make here is that we can definitely start to make some assumptions about the shape of the molecule in this case. And we can extract some structural parameters such as the radius of gyration, some compactness, the maximum length scale, and shape just from doing this analysis. But again, it requires that you have that monodispersed ideal sample. So let's look at a few examples. So we're gonna look at first something for glucose isomerase. So this is a concentration series that was collected in our laboratory for protein solutions that are 4 milligram, 2 milligram, and 1 milligram per mil for both 3 minutes and 5 minutes. And what we see here is if we look at the Guignay plot, then it's fairly consistent around 30 to 31. And the Guignay, the I0 versus C, which remember that's proportional to the molecular weight, is fairly consistent. But there is a slight increase in this case. We can also look at molecular weight and radius of gyration from some of the other analyses that we do, for example, the pair-distance distribution function, and we can look at the maximum length scale, which is around 92 to 94. So all of these are quite consistent with glucose isomerase. We do have some variation within these, but what we can say is we're very close to visualizing the form factor scattering for this molecule. And once we do that, we can start to calculate some models for what it looks like. So the red on the bottom is the actual structure of glucose isomerase from the protein data bank, overlaid with some calculations of what the expected shape would be based upon the P of R function that we see. So this is an example of looking at a highly diluted solution. What happens if you can't ever get to the form factor because you see some sort of, or if you need to look at both high concentration and low concentration? Well, we did an experiment like this for monoclonal antibodies. These are used as therapeutic agents, and ideally you want to administer them at high concentrations because that means you administer lower volumes. But you also want to make sure that you have good stability and you don't administer any agglomerates or aggregated material. So the question was asked, we're going to look at 2 monoclonal antibodies at a concentration range from 1 to 100 milligrams. And we're going to ask, can we ask questions about the form factor, the structure, because we want to get to the form factor, but we also want to look to see, is there aggregation or repulsion? And we should be able to tell that. So here are the observed SAXS profiles shown here. And in this case, what we do see is for one of the monoclonal antibodies, as you increase the concentration, you see that there's a downtick intensity at the low scattering angle. And alternatively, the opposite is true, although to a lesser degree for the second monoclonal antibody. But what it shows is that both samples deviate at low scattering angle. And if we zoom in and take a closer inspection, it shows that the intensity is decreasing as the concentration increases in this case. So we'll see here that it's dropping down as this concentration decrease. And this is that hallmark of interparticle repulsion that I talked about. You know, they are distinctly different between the two of these. So then we can ask the question, can we start to extract information about the solution structure factor and ask some other questions? In addition, we can see that at the lower concentrations, there is some stabilization of the two plots. So we can assume that, or presume rather, that these lower concentrations are a true representative of the form factor. So if we do that, then we can take the data and do the indirect Fourier transform and look at the P of R plot that tells us the maximum length scale is 150 to 200. We can calculate some models, and then we can ask questions about how closely do they fit known structures for these monoclonal antibodies. So that's something that, that can clearly be done. All right, but can we say something about the solution structure factor? Well, absolutely we can. What we can do is we can solve for that component by dividing the intensity of an experimental data set by the form factor, basically the data at low scattering angle, this right here. We can extract information. If we do that for each of these scans from 5 to 100 milligrams, we see their solution structure factor values. And basically, it's decreasing as concentrations increase, but the intensity can also be different for different scattering angles in this case. So for example, this particular monoclonal antibody only exhibits repulsion, whereas this one exhibits both a bit of repulsion and attraction. So we can start to say something about those behaviors. Well, what about even more concentrated solutions, say crystalline suspensions? And these are quite often used in pharmaceutical applications. And in this case, we can start to ask questions, is it amorphous? Do we have a crystal presence? And if we do, what particular crystal parameters do we have? And we can do that just by putting our solution in the beam. Something that's crystalline is going to start to exhibit some X-ray diffraction light behavior, and something that's not is going to look more like an aggregated protein solution. So we did that for a few samples in our laboratory. So these are human growth hormone. And what I wanted to just show you are the results of those experiments quickly to show that for the few of these that we did, we could clearly identify which were crystalline and which were not. And we could also identify those that were non-crystalline, and they showed this different slow change in intensity over scattering angle. And because we have this and we have the peak positions, then we can start to identify their crystal forms as well and extract information and perhaps start to manipulate these solutions to push them one way or another to a specific crystal condition or not. So with all of that, I hope what I've showed you is that it's absolutely true that we can quickly evaluate protein structure using small angle X-ray scattering. We looked at some of the information we can get from SAXS. We looked exactly about how does SAXS work? What does the experiment look like briefly? We looked at dilute versus high concentration solutions and the things we can extract. And then finally, we looked at an ordered molecule. So with that, I'd really like to tell you how much I appreciate you joining us today. And if you have any questions, I'm happy to take those now.